Lynn, I'm turning it over to you.
Okay. So good afternoon. My name is Lynn Heiler, and I work in the Children's Center for Undergraduate Excellence. I am located in Funkhauser in what we call Funky Town, but it's good to be with you today, wherever you may be. This presentation is being recorded just so that everyone knows, and our audience is far and wide, and it will be permanently located in the cloud. We will have a question and answer session, and if you will just use your Q&A button at the bottom of your screen, when we get to that point, I will either call on you or your presenter can do that as well. So we are going to get started with Shelby, who is our first presenter, and you should be able to share your screen, Shelby.
Hi, everyone. Thank you for being here today, even though I know many of you have to. All right, are you all able to see that okay? Okay, perfect. Thank you for the introduction. My name is Shelby McCubbin, and I'm a junior here at UK, studying neuroscience with a minor in gender and women's studies. The research that I will be sharing with you today is titled Stretch-Activated Channels Maintain Heart Rate in Larval Drosophila, and it was conducted under the mentorship of Dr. Robin Cooper in our department of biology. I'm going to go ahead and walk you through an introduction and some previous research, followed by what we're currently working on, and ending with a few minutes for further questions. So to start off, let's talk briefly about Drosophila. If you've ever taken a biology lab course or genetics, you're probably pretty familiar with them already. They're also known as fruit flies. They are considered to be one of the most common biological models in research because they're both easy and inexpensive to use. They have a short life cycle, and they can be genetically modified in a number of ways. Additionally, their hearts are structurally similar to many mammalian models, but they have a simpler physiology. These Drosophila hearts can be dissected and examined through different bioassays to study a large variety of problems. However, we quickly run into the issue that heart preps are not long-lasting, and when we examine a heart prep in a static bath, the heart rate is going to drop throughout. In fact, we see a devastating loss of function about 2 hours in, which is going to prevent the use of long-term stimulation. What's really interesting, though, is that we see a partial restore of function with the addition of saline, which kind of brings the question of what effect a stretching device would have on the maintenance of heart rate, because the saline is being administered through a syringe via this kind of flushing movement of the bulb. So here you can see a heart stretching device. It's made up of speakers and a stimulator. And so what's happening is magnets cause the speaker to have these slight movements that cause an attached pin to stretch and relax the heart. And so when you look at the graphs on the right, this top row shows a set of preps in a static bath. And you can see these really dramatic peaks and falls, followed by an exhaustion of the prep at the end. These graphs on the right are just an average of all the different preps. The use of the stretching device in the bottom row, however, results in not only less dramatic drops and peaks throughout, but also a higher average % heart rate at the end of data collection. This next slide includes an image of a continuous perfusion pump, which is going to have a flow rate of 1.6 milliliters per minute. So that speaker from the last slide is a great improvement over a normal static bath. But the heart tissue would usually be torn by the end of those 2 hours. And so this perfusion setup is a lot better because it still gets that saline moving, but there's less overall stress on the prep. And the solution can also be easily switched out, which is nice. And so sure enough, you can look at these graphs on the right again. These two are the exact same ones as these in the last slide. But this new bottom row is the data from the continuous perfusion pump, which you can see that those dramatic peaks and peaks and drops are no longer there, and we're still maintaining this high average % heart rate. So this begs the question as to why this stretching is beneficial for heart rate mediation in general. And the best, or at least most obvious explanation that we have, is that there may be stretch-activated ion channels present in the Drosophila heart. And this would make a lot of sense because we know there are stretch-activated ion channels present in mammalian hearts, as well as in Drosophila sensory neurons. We also know that there are TRPA channels present in the heart, which are known to be sensitive to temperature, but they may also be sensitive to mechanical stress or the stretching. And so last semester, the Bio 446 class here at UK conducted a panel of pharmacological agents to try and identify the stretch-activated channels in the Drosophila model. And the work that they did suggests that the heart rate stabilization may be due to a subset that's sensitive to amiloride, gadolinium, and GSMTX4, which is a tarantula toxin. So from there, we looked at heart rate regulation and a knockdown of piezo in mesodermal originated tissues, because piezo channels are mechanosensitive, and mesodermal tissue is going to make both the skeletal muscle and the heart in the Drosophila system. And so we saw no significant difference in heart rate in these RNA knockdowns, which is going to suggest that those stretch-activated ion channels in Drosophila must not be piezo-activated. Otherwise, we'd have expected a response in these preps. However, there are still many different channels that could be responsible, so we are conducting a proof of concept to see whether the TRPA receptor is causing an effect by overexpressing it in the heart, to see whether this overexpression improves pulsatile influence in maintaining heart rate. And this really goes back to the beauty of using that Drosophila model like I was talking about in the beginning, because of how easy it is to manipulate these genetic changes. And so the reason we're looking into the overexpression of TRPA1 receptors specifically is that we see an increase in TRPA1 expression in defective mammalian hearts, but it's unknown currently whether this change is a beneficial change to help mediate problems as a type of compensatory mechanism, or whether it's actually a pathological response to disease. So if the overexpression of TRPA1 receptors in this model helps maintain heart rate, that could kind of suggest that the increased TRPA1 expression in human pathological states could actually be a beneficial response. So we're currently in a state of data collection, but have not completed any data analysis yet to see whether that overexpression is in fact making a difference in maintaining heart rate. We have taken 2 different parental lines and crossed them to create an F1 generation as well. Additionally, with the cross generation, we are applying the pharmacological agent HC030031, which is a known blocker of TRPA1 receptors, to see whether that blocker has an effect on heart rate as well. Because if the overexpressed TRPA is able to better maintain heart rate, the blocker should kind of give us opposite results. Lastly, these preps are being looked at in 2 different temperature conditions, both 25 degrees Celsius, which is kind of like our scientific room temperature, as well as 30 degrees Celsius. And this is because, again, those TRPA1 receptors are known to be sensitive to temperature. So we want to look at different temperature conditions throughout the experiment as well. So that's really as far as we've gotten for now. I'm hoping to move forward with data analysis as soon as we all survive finals, and hopefully I'll be able to pull together some answers throughout the summer, but I'm happy to take any questions you all may have.
Shelby, there is one question in the Q&A box. Can you see that? If not, I'll read it to you.
Yes, it took me a second to find it, but I can see it now.
All right, go ahead.
Okay, yes, the question is asking whether in the first couple of graphs, if the graphs on the right side were the average of the measurements of the graphs on the left. I'm gonna flip back for you so you all can see what we're talking about. But yes, these on the left are, I think, 6 different preps, and so the ones on the right are just an average, so you can see the overall trend.
Hey, Dr. Cooper says fantastic presentation. And then his statement or question is, I wonder if we keep the heart saline a bit cooler, if it would last longer with a slower heart rate to start with, maybe lower metabolism for non-TRPA1 preps. Just thinking out loud for the next projects, hint, hint.
Dr. Cooper is always trying to stem off these different projects from what we're working on. But it would definitely be interesting to look at lower temperatures as well. We have another undergraduate in the lab named Nicole who did a lot earlier this semester and last semester with temperature sensitivity, but she was increasing the temperature. So an interesting thing we saw is that it always cut out around 37 degrees Celsius. It was too high for the heart to maintain. But I don't think she really looked at lower temperatures. So that would be interesting for us to look into.
And you have a couple of comments that says, great job from Stats Guy, and that he is from Chicago, logging in and watching today. So welcome.
Thank you.
So Shelby, tell me how you got involved in research.
So I got connected to Dr. Cooper through a mutual acquaintance a couple years ago. And so I started out in his lab my sophomore year. And we started off with a little bit of a simpler project then, but I think he's a really great mentor and I really enjoyed the lab environment. And so I've kind of just stuck with him throughout the past 2 years, and I'm going to keep bothering him next year as well.
I'm sure he won't mind.
I hope not. Jesse, I see your question about a challenge that you encountered. I think COVID has been the biggest difficulty for everyone. It's been really hard for us to get into the lab. Right now, Dr. Cooper has been running some preps, and I've been kind of looking at the data asynchronously, like from my laptop, whereas in the past, I would get to actually go into the lab and do the research with him. So we've made it work, but it's just been kind of disappointing to not be in the middle of the action as I normally would. But we're hoping to kind of solve that a little bit over the summer.
You just have a few comments in there for yourself. Keep him busy, they say. Glad you're coming back.
Always.
Okay. I don't believe there's any more questions, so we will come on to the next presenter here. Thank you, Shelby.
Thank you.
So Cecilia, if you're ready, you may start anytime. Share your screen. Thank you.
Thank you. All right. So hello everyone, my name is Cecilia Pankau and I'm currently a junior at UK majoring in neuroscience and minoring in Spanish. Today I'll be sharing with you my research pertaining to the development of an authentic course-based undergraduate research experience, or ACURE approach, in order to study the effects of manganese on physiological processes. My research was conducted under the mentorship of Dr. Robin Cooper within the Department of Biology. I'd first like to start off by giving you all information about the ACURE approach, which has been developed to educate students within STEM. The ACURE approach was based off the CURE approach, or course-based undergraduate research experience, which has been developed previously. The primary difference between these two approaches is that the ACURE approach emphasizes the facilitation of a more reliable research project and realistic, involving unknown outcomes. Utilizing this ACURE approach, we investigated the effects of manganese on the physiology of various invertebrate species. Because literature has indicated that overexposure to manganese can cause Parkinson's disease-like symptoms, acute cardiovascular disorders, and other symptoms, we investigated the effects on larval Drosophila melanogaster cardiac function, heart development, behavior, and sensory neural function in crustaceans. Because our work integrated various themes of investigation into one cohesive project, our experimental design was relatively novel and promoted a multifaceted understanding of our research. This course integrated a class of freshman STEM students once a week, and the course was conducted with the students presenting in the end within the UK Undergraduate Research Showcase. Now moving on to our research. The first graphs depict research studying the effect of manganese on cardiac function in Drosophila melanogaster. When exposed to 0.1 molar manganese sulfate solution, the heart stopped and one is unable to restart after the manganese was washed out. In contrast, the hearts of larvae stopped when exposed to 0.001 molar manganese sulfate, but the heart was actually able to restart after the saline washed the manganese out. Next, moving on, we look to investigate the effects of manganese-treated cornmeal on larval Drosophila melanogaster body wall movement and mouth hook movements per minute. We investigated the effects of both manganese sulfate and manganese chloride, and this was an idea that was introduced by students within the class. As shown by the graphics, body wall movements per minute decreased significantly when exposed to 0.001 molar manganese chloride cornmeal, but increased slightly when exposed to 0.01 molar manganese chloride. Additionally, larvae exposed to 0.001 molar and 0.01 molar manganese sulfate had decreased body wall movements. However, 0.001 molar decreased to a greater extent than 0.01 molar manganese sulfate. Additionally, there's a significant decrease in mouthhook movements per minute when fed food containing 0.001 molar manganese chloride, but the difference was not significant when fed 0.01 molar manganese chloride. Similarly, when fed both 0.01 molar and 0.001 molar manganese sulfate cornmeal, the number of mouthhook movements decreased slightly. Next, we investigated the effect of manganese on sensory neurons within the blue crab. The graphs on the left depict the change in extracellular field potential of a sensory nerve when exposed to various solutions. However, the graphs on the right depict percent change and demonstrate that preparations exposed to 1 molar manganese sulfate had the greatest percent change within the experiment. The next section depicts a similar experiment, except with crayfish. We had intended to complete this experiment. However, unfortunately, we did not have enough time to do so. But moving forward into the summer and once finals are all done, we hope to do this experiment in the future in order to expand our research further. The final graphics to the upper right demonstrate the effects of manganese on the survival of Drosophila melanogaster as they developed from first instar to pupa. And this development is shown in the diagram in the upper right. The study showed that there seemed to be no significant differences as when exposed to manganese sulfate and manganese chloride. Moving forward, we plan to utilize this project as a guide for potential research through the investigation of new research questions and the search for deeper understanding of topics that have already been addressed. Working with lower level students required time to explain concepts investigated during the class. However, this extra effort led them to be confident enough to present in a university level research conference. We believe that utilizing the ACURE approach provided students with a realistic perspective of scientific research in the process. And this sparked an interest in many of them to hopefully lead them to further their involvement within research in the future. Thank you for all of your time. So please let me know if you have any questions related to the ACURE approach or any of the research that has been done during this time. Thank you.
Thank you, Cecilia. That was a wonderful, wonderful presentation. Very good. Okay, can you see the questions there? Or would you like me to read them?
Let me see. All right. Yeah, okay, I found them. In the first couple of graphs were the set of graphs to the right, just the average of the measurements on the graphs to the left, just asking because I couldn't read the y-axis. Yeah, it was a really challenging to fit the graphs on here, so I apologize for that. However, first couple of graphs. So the graphs shown on the left are the body wall movements. And the top left graph shows the body wall movements when exposed to manganese chloride. The bottom left graph shows the body wall movements when exposed to manganese sulfate. And on the right, we are showing the mouthhook movements. And the same shows with, on the top, there's manganese chloride, and the bottom is manganese sulfate. So I apologize for how difficult it is to read, but the left column is body wall movements and the right column is mouthhook movements.
I think that's good. Thank you.
Mm-hmm.
Now, Dr. Cooper asked, so what was the most challenging part of directing a whole class of students on a research project?
I'd say the most challenging part was going into each class and trying to explain concepts to them, and within that same class, you'd help them facilitate the research done. So it would be fantastic if we had more time to do the research with them in order to reduce the variability of the experiment. However, that was challenging to do so because not only were we explaining how to do the research, we were also explaining concepts behind it. Because as lower level students, they're not entirely familiar of concepts that I learned in Intro to Neuroscience or other classes such as that. So I feel like it was just challenging because there was so much information to put together into one class period.
Okay, and then he says, so what was the most rewarding part of the experience?
I would truly say that like making the connections with the students was one of my favorite parts, because they're all like very passionate students about the research and super interested in what we were all doing. And I found like I had a great connection with the students and served as more of a mentor to them, which was really rewarding for me personally.
Great, great. So I don't think there's any more questions in the Q&A, but just for my benefit, why don't you tell me how you got involved in research, please?
Yeah, so I actually, I started my undergraduate career as a nursing student, and I did that for a year and a half and ended up changing my major last year, so last spring. And COVID happened. It was all like me trying to realize I majored in neuroscience and wanted to get involved in so many different things because I wanted to go to medical school. So it was kind of a rush to figure out a research mentor, but I ended up actually having a Dr. Robin Cooper is an academic advisor, and I ended up having a meeting with him this past October. And we just had, I was asking him about questions about my schedule, and that just led into so many conversations about like med school, advice for med school, his research, and how we bonded over a love for optogenetics and a bunch of different other things. So it kind of just led to me asking him if I could join his lab. And now I'm in his lab, and I have enjoyed it so much. It's been very rewarding.
That's really good. Thank you. So you have one last question here. It's from Douglas, and he asks, were students conducting any activities outside of the lab, and how much time did they spend outside of class time?
So as far as research, they did not conduct any research outside of lab. That was done by Dr. Cooper and I. We supplemented the research that was done within class. However, when it came to the UK undergraduate research showcase, that involved some time for them to create their abstracts and their posters. So those who did the presentations did have some extra class time involved with that. However, research in itself was done by Dr. Cooper and I outside of class time.
Thank you very much. It was a wonderful presentation.
Thank you.
Mm-hmm. So our next presenter for the day is Alexis Taylor. Alexis, you may start anytime and share your screen as you see.
Okay, well, good afternoon, everyone. As that was just mentioned, my name is Alexis Taylor. I'm currently a junior here, and I do research in the Department of Biology under the direction of Dr. Osborne. Dr. Jeffrey Osborne, as well as some of our graduate students, Shay Sickles and Chelsea Weaver, who is now Dr. Weaver. She just defended a couple weeks ago, so congratulations to Chelsea, as well as with Dr. Jessica Taylor, who works in the Department of Physiology and Biophysics at Case Western Reserve University in Cleveland, Ohio. And so the research that I do focuses on identifying genetic loci in the African green monkey that associate with hypertension in a purpose-bred F2 generation offspring. So kind of a mouthful. So hopefully I'll, through our conversation, I'll be able to break that down a little bit for you. So first, just some background. So why hypertension? So hypertension and other cardiovascular diseases are the leading cause of death nationwide. So right, we know this would be even more of a cause for death than even cancer. And the animal model that we use is the African green monkey, which is very unique, and that is the only non-human primate that develops hypertension similar to us as human primates. So this can either be through their diet, so if they have a diet high in salt, fat, or high cholesterol, or through genetics. So we know that if their parents experience hypertension, they're also more likely to experience hypertension. And we see the same things in us as humans. And so we also do this work in conjunction with Primates Plus, which is a research colony down in the island of St. Kitts and Nevis in the Caribbean that maintains a significant African green monkey colony used for scientific research. We're one of the only ones in the world and one of the biggest at that. So we're very proud about that. And so the main goal of my research is to use, by using genome sequencing in a purpose-bred African green monkey colony, we can determine genetic quantitative trait loci that may correlate to human essential hypertension. And so the methods that I use mostly to do this work is, first, we consistently measure our systolic and diastolic blood pressures and heart rate in all of our monkeys that are in our breeding groups. And we try to do that about 3 or 4 times a year, every single year, to get good, consistent measurements. So we have male and females that are phenotyped as hypertensive, and we phenotype monkeys as hypertensive if they have a consistent systolic blood pressure of at least 140 milligrams of mercury, or normotensive, which is systolic blood pressure of, again, a consistent systolic blood pressure of less than 140. So we also have offspring now, which is very exciting. So Primates Plus was established in late 2015 with our first breeding group, happening in 2016. So now all of our offspring that were born in 2016 and 2017 have reached sexual maturity, and now we can use them to generate F2 breeding groups. So that's kind of where I come in. And so I have now been tasked and work on creating these specific breeding groups based off of the phenotypic data, if they're identified as either hypertensive or normotensive. And so I use pedigree charts mostly to track our lineages and recognize specific offspring to assist in generating the F2 generation breeding groups that we have. So what I have right here is just a simple bar graph to show you what kinds of breeding groups we currently have in our monkey colony. So a mix of different normotensive males, normotensive females, normotensive males with hypertensive females, hypertensive males, normotensive females, and hypertensive males and hypertensive females. So as you could probably guess or assume, this hypertensive breeding group was one that we really looked the most at, right? And so down here, like I mentioned, I'm working on creating an F2 generation. It's a big task. So these are some of the pedigrees that I've started working on for this F2 generation. So we have 2 different breeding groups. As you can see, our breeding groups are made up of 1 male with several females. The squares represent males and our circles represent females. These red, if they're in red, that means that we phenotyped them as hypertensive. So these have all come from this hypertensive breeding group, right? Because as I mentioned earlier, genetics plays a huge part in this. So if both of your parents are hypertensive, you're probably hypertensive as well. The ones that are white are ones that haven't been phenotyped yet, right? So due to COVID, we have not been able to get down to St. Kitts and Nevis, right? It's in the Caribbean. It takes a little bit to get down there in order to phenotype these animals as much as we would like in order to confidently phenotype those. So that's something we hope to do in the future, is be able to get more measurements so that we can phenotype them. So some of our conclusions are, again, the African green monkeys, what we use to study our hypertensive our hypertension studies. We have specific breeding groups made up of hypertensive males and females, and that's how our colony is organized. So once our offspring reach sexual maturity, new breeding groups can be created for an F2 generation, which again is what I've been working on these past few months. We collect tissue samples when we go down there, so that way we can analyze possible genetic loci that could correlate for hypertension. And once we can do that, we can then compare them to other species who experience spontaneous hypertension, like human primates like ourselves. So future directions. So again, I've planned out all of these F2 breeding groups. So they will now start to be introduced in our African green monkey colony. So it takes the monkeys a little bit of time to kind of get to know each other a little bit when they are first introduced into the breeding group. So we're working on getting those introduced now. And then just like with all of our other monkeys, these breeding groups will get consistent systolic and diastolic blood pressure measurements and heart rates, as well as potential pregnancies. So we are now in the prime of the mating season. Mating season will go all the way up until about end of September, early October. So a couple times a week, we have to check all of our females to see if there's any pregnancies that we then need to monitor and keep track of. And then again, with our tissue samples, we do DNA extractions in order to whole genome sequence to identify our genetic loci for hypertension. And that again helps us to try to correlate to further advance the hypertensive studies to help the human population. So that's all I have for you. Again, I would like to thank so much all of my research mentors and all of the other undergraduate students that are in my lab, as well as the staff down in Primates at Primates Plus, who do a great job of taking care of our animals, our research subjects down there. So again, thank you all so much for being here. I'm more than happy to take any questions you all may have.
Thank you. Great job, Alexis. So I do have one question for you already in the queue, and it is from Jessie. And she says you did a great job. If you had a do-over,
yes,
is there anything you would do differently?
Right, yes. So definitely try to start phenotyping these a little bit earlier. We actually had a little small hiccup. I had all my pedigrees nice and organized, and in the software we were using, the company no longer decided to sponsor it. So I had to kind of completely redo over all of that. So that gave me a little bit of hiccup. So starting that over earlier, that way I could try to get these 2018 or these 2016, 2017 breeding groups. That way they could be introduced before mating season happened. That way they could kind of get a feel of each other before mating season. So again, now we're kind of pushing it to see if we can get any pregnancy. So yeah, I can get a little bit more on top of it if that's possible.
All right, that happens. Okay, and then Dr. Cooper says, nice presentation. And where do you get the DNA samples from on the monkeys?
Yeah, so we get them from a whole bunch of places. Ones that I usually use are just from ear samples. So we take little notches of their ear and are able to from there. And again, we also have different tissue samples. So a girl in our lab does one, she does brain, we also have lungs, stuff like that, but I use mostly ear notches.
Let's see if there's any more questions that pop in there. We'll just give it just a second or two. There's a bit of a delay sometimes. Looks like you're all good. No more questions for you. Thank you, Alexis. You did a great job.
Yeah.
Okay, so our next presenter is Olivia. Olivia, are you ready to go?
Hi, I am.
Okay. You may share your screen at any time.
Okay. Can everyone see that?
Yes.
Okay. My name is Olivia, and I'm a neuroscience major at UK. My faculty mentor for this project is Dr. Mark Prendergast. And I would also like to say a huge thank you to Jessica Gabhart, one of the recent master's program graduates, for being a mentor to me on this project. So the topic of this project is fetal alcohol spectrum disorders. And fetal alcohol spectrum disorders are cognitive disorders that arise from exposure to ethanol in utero via maternal drinking. So if you were to look across a group of 100 first graders today, you would see that 1 to 1.5 have a fetal alcohol spectrum disorder. In the fetal brain, exposure to ethanol triggers reactive oxygen species production, lipid peroxidation, a decrease in AMPK, and an increase in neuroinflammation and neurotoxicity. So within this pathway, we are especially interested in the role of AMPK. And recently, animal models of neuroinflammation and similar metabolic disruptions have shown that ICAR, which is a synthetic AMPK activator, can have a neuroprotective effect by regulating AMPK. So this study asked the question, can ICAR have these same neuroprotective effects in a third trimester binge drinking model of fetal alcohol spectrum disorder? So to test this, what we did is culture slices from 10-day-old rat pups, which is the developmental equivalent of the third trimester in humans. And we set up 4 different groups. The first group was a control, which only received plain media. Second, we had the ethanol group, which was exposed to 50 millimolar ethanol for 5 days, and that imitated ethanol exposure in utero. That amount of ethanol is the equivalent to a blood alcohol of 0.23, which is 3 times the legal limit. So this model would represent a binge drinking episode. Third, we had the ethanol plus ICAR group, which was exposed to ethanol for 5 days and then exposed to ICAR. And that would simulate binge drinking followed by this ICAR containing drug. And finally, the ICAR group was our positive control. So we stained the slices with propydium iodide and used fluorescence microscopy to analyze the amounts of cell death after 24 hours in 3 different regions of the hippocampus, the CA1, CA3, and dentate gyrus. And these images right here represent the way we measured cell death using the PI. The darker area shows less cell death, while the brighter areas show more cell death. Using images like the ones here, we measured the amount of cell death in image J, and our results showed that in the CA1 and CA3, the ethanol groups showed a greater increase in toxicity than the control, while the ICAR group showed a decrease in toxicity compared to the control. The ethanol plus ICAR group was not statistically significant from the control, and the ethanol group showed a greater increase in toxicity compared to the ethanol plus ICAR group. So all in all, this shows that ICAR is protective against ethanol-induced neurotoxicity in the CA1 and the CA3. Now, the results from that third region, the dentate gyrus, showed the same conclusions, but in this region, we also found that ICAR was protective to an effect beyond the control. This difference might be explained by the different cell types in the CA regions versus the dentate gyrus. In the CA1 and CA3, you have pure metal cells, while in the dentate gyrus, you have granular cells. The CA1 may be especially sensitive because it contains a high density of NMDA receptors. And overall, we concluded that in all 3 regions, our hypothesis was confirmed. We showed that ICAR is protective against ethanol-induced toxicity. And this experiment is currently being replicated to account for possible litter effects. As far as future directions, we want to confirm the PI imaging with immunohistochemistry using NUIN, which is a marker of cell life, whereas PI was a marker of cell death. So NUIN is a reliable marker for mature neurons. It stains FOX3, and we want to see that the NUIN results are inversely correlated with the PI stain. So in conclusion, the takeaway is that right now there is no treatment for fetal alcohol spectrum disorders, but this preliminary study shows proof of concept for using ICAR to actually reverse the neurotoxicity in the fetal brain and prevent the lifelong consequences that come with fetal alcohol spectrum disorders. So that is all, and I'm happy to take any questions.
Thank you. That was really good, Olivia. Appreciate it. Let's see, there's no questions yet. So there's a little delay sometimes. Let's tell us how you got involved in research, and has this affected or encouraged what you're going to do in your career?
Yeah, so I actually started working at a molecular genetics lab when I was in high school through UK, and really just fell in love with RNA processing and the project I was working on there. And then once I kind of maxed out all my research hours in biology and needed to focus on neuroscience, I got involved with this project actually under Jessica Gabhart, one of the graduate students. And then I actually followed her this semester to start working at Naprogenics, which is a biotech company that has a lab on campus.
Let's see. Reset it sometimes if they don't pop up there right away. Okay, here's one. So Dr. Cooper asked, well, first of all, he says, great presentation. And then he says, do you think the toxicity is due to high calcium Thank you. Go ahead, intercellular. That's it. I was trying to put it all together there.
That is definitely a possibility. The pathway that we kind of put together, you can see here, what they think happens in the papers that I've read is that ethanol induces reactive oxygen species, and then that creates the lipid peroxidation and then the decrease in AMPK. But there's definitely, I mean, as you know, like a lot of kind of side pathways that would influence that, and high calcium concentration could definitely be one of those.
So, and the next question is, what challenge did you encounter, if any, and how did you overcome them?
I think kind of what some of the other girls have said, with COVID, we weren't necessarily allowed to be in the lab. So a lot of it was remote work. And for this project, this semester, a lot of it was just having the data and learning about it without actually being in the lab. So that was difficult. And I think I overcame that just by reading and getting involved in every way I could, minus actually being in the lab.
Dr. Cooper just made a comment. I'm not sure if you can see it. He says, thanks. Thinking ROS might cause mitochondria to leak calcium.
Yeah,
Right?
that makes a lot of sense. Yeah.
Well, thank you very much, Olivia. Great job.
Thank you.
So our next presenter for the afternoon is Grace Jacobs.
Okay, I'm ready. Let's see here. Can you see that okay?
We can, yes.
Awesome. All right. Okay, so I'm good. Hi, everyone. My name is Grace Jacobs, and I'm currently a sophomore here at UK. I work in the Cooper Lab under my research mentors, Dr. Cooper, Dr. Kay, and Dr. Madu. And today I'm going to be talking to you about my project on the social interactions of the Charax quadricarinatus. So the Charax quadricarinatus, commonly known as the Australian red claw, is one of 3 Australian crayfish species that were present in aquaculture in 1997. They were really the ideal candidate for expansion in the aquaculture industry because they can tolerate a wide range of environmental conditions, and they have a much larger size than the crayfish that we are accustomed to seeing and eating in the United States. So again, we can see here that 27 tons per year on average since 2010, they've been increasing in that aquaculture industry. And while it might be good for the industry, it could have unintended ecological impacts because crayfish are actually pretty tricky and they can easily escape from the aquaculture facilities, and as well as personal crayfish usage not being tightly controlled. Either of those types of introductions could lead to competitive interactions with other widespread native species. So we decided to investigate the behavioral nature in intra-species pairings, as well as inter-species pairings with a well-established aggressive native crayfish. I hypothesized that the Australian would actually not be an aggressive species with itself or with others. So to give you a little visual here, these are the 2 species that we're working with. This one here on the left, you can see is a large Australian red claw or a large Charax quadricarinatus, and the aggressive native species selected for this project on the right here is the Faxonius virilis, commonly known as the northern crayfish because it's native to northern parts of the United States. Procedurally, all crayfish were placed in isolation for about 2 weeks prior to any interactions. Then that was just to help ensure that they didn't remember any of the previous encounters that they had that might impact their performance in that pairing. For the pairings themselves, we had 2 size-matched individuals placed in a novel tank at the same time and recorded on video for about 20 minutes. From there, each individual's interactions were scored from that video footage, like I said, and we are scoring based on a 4-point scale that was kind of first published in a paper by Jimenez and Fox in 2011. So fun little video clips here. We've got a 1 on our 4-point scale, which indicates that the individual interacted with no aggression. So as you can see here, we've got this front crayfish kind of bumped into the back crayfish and walked away. So both of those individuals would have received a 1 in that scoring methodology. A 2 is going to be threat displays. So you can see here, this crayfish on the right is actually producing a threat display towards the crayfish on the left. So this right-hand crayfish would receive a 2 on our 4-point scale. A 3 is where it starts getting a little dicey. This indicates a claw lock, and this is when one individual is actually going to grab onto its opponent. So as you can see, both of these small Australians have quite a good grip on each other. So both of them would actually receive 3s in that scoring system. Finally, we have a 4. So a 4 is going to be a strike or a strike and rip. And this is one individual making unrestrained usage of its claws. So we can see this kind of tanner colored crayfish will actually rip off the leg of this blue crayfish right here. There's that leg kind of floating there. So this tan crayfish would receive a 4, and that is the highest level of aggression on our scale. The program that I use to kind of track all that scoring and keep everything neat and tidy is called JWatcher. It is a quantitative analysis tool put out by UCLA, and I've linked it here if anyone's interested. It can be freely downloaded off their website, and it really enabled my scoring process to be much easier than doing everything by hand, much more precise. It also allowed me to standardize my methodology so that my procedures can be repeated in the future, which is exciting. Another fun little kind of side note here is that it helped me continue with my research during the pandemic. So as some of the other presenters in here are talking about early on, not being able to be in the lab can be pretty tricky. And so this was a fun and creative way to kind of circumvent that problem that came up. With the way we're actually going to use JWatcher, I've programmed single alphanumeric keys to correspond with each of the behaviors. And at the end of 20 minutes, it'll give me this nice time-stamped printout with all of my behaviors, as well as a summary table of each individual's data. So the current results of this ongoing study can be seen here. We've got the small Chiracs on the right-hand side, and we've got the large Chiracs on the left-hand side. And frankly, the small Chiracs appear to be more aggressive than the large Chiracs, with around 83% of the small Chiracs reaching a 3 or a 4 scoring, whereas only 29% on average of the large Chiracs here receiving a 3 or 4. The intraspecies pairings of the Faxonius virilis and the interspecies pairings are still in that scoring phase. So while I don't have those results today, I'm really looking forward to seeing those outcomes in the near future. Finally, I'd like to thank my mentors, Dr. Cooper, Dr. Kay, and Dr. Madu, as well as graduate student Allison McLaughlin for helping me, encouraging me, and introducing me to JWatcher. I'd also like to thank Dr. Schuster for species identification and Dr. Nadolski, who will be helping with data analysis. Many thanks for everyone listening, and I'm happy to take questions at this time.
Thank you, Grace. That was wonderful. You did a good job. While we're waiting on a question or two to pop into the Q&A box, would you please tell me how you got involved in research?
Yeah, so I actually started research with a Bio 199 class in Dr. Famolsky's lab pretty early on. And with the height of the pandemic and Dr. Cooper being my neighbor down the street, reached out to me and said, hey, I've got a really cool project that I think you would enjoy. Let's get started on it. So that's kind of how I came into this project, and it's been awesome working with him and all of the other mentors that I listed there. So yeah.
Okay, thank you. We do have a couple questions for you. Well, one question and one comment. So Dr. Cooper says, great presentation. And how practical do you think it might be to use a citizen science project using this software now, seeing the variability STEM students have?
Yeah, so he's kind of referring to, we actually were able to take that, as I kind of mentioned, the reproducibility of the JWatcher and apply it to a STEMCats class, one of his spring STEMCats class. I think it is a good thought and theory. I think JWatcher itself needs to be updated, modernized, for it to be practical for more of the general public to participate in it. But I think if you were in an educational setting like this one, or even in a high school classroom, and you had the ability to sit down and walk someone through the J-Watcher program, it would be a lot more feasible.
And that was the second part of his question. Jeremy says it's a little biased, but a great presentation. He's a little biased, but a great presentation, I think. And then Jesse asked for the aggression scoring process. What was most challenging?
I think the thing, let me see if I can pull up one of my images here. I think the thing that was most challenging, as you can see with this one, so with the bigger crayfish, we obviously had to keep them in bigger tanks so that they had room to move around. Sometimes the water, which we did change between either every interaction or every 2 interactions, I believe, could get a little cloudy when they're walking around in the sand. So this is a good example of it being not the clearest video. Some of them were even more fuzzy and harder to see. So we actually went back and re-recorded some of the interactions from a top-down angle, which is like this angle, because that was a problem.
I see. Okay. Right. So I don't believe there's any more questions for you, Grace, at this time. Thank you for your presentation. Very
Awesome.
interesting. So it looks like our last presenter of the day is Anna. Anna is with us. Anna, you can start sharing your screen anytime you'd like. And just don't forget that you're muted. Okay, thank you.
Okay, hello everyone. My name is Anna Hawse. I'm a biology major and philosophy minor, doing philosophy research with Dr. Kirchner. I've spent this past semester looking at the different philosophical approaches to animal ethics. This is a really big area to cover because it includes everything from our pets to wild animals. And while I found that the ethical theories sometimes touch on our domesticated animals, they rarely address the wild ones. And we tend to disregard how much we impact wild animals. So to illustrate the degree of the problem, if you'll look at my first slide, this is cane. It's a kind of native bamboo that used to be really common in this area. The cane fields would stretch on for miles. But now you can consider yourself lucky if you've ever seen any at all. And it's pretty clear why, because it was virtually eliminated when this area was settled. Next, this is a chestnut tree. Like the cane, you can consider yourself really lucky if you've ever seen one, because virtually all of them were killed by chestnut blight, which is an invasive disease from Asia. They used to make up a massive portion of eastern forests, including in this area, and supplied lumber to humans and food to both animals and humans. They're both plants, but I think these demonstrate really well how profoundly we've altered our environment, even here in Kentucky. And this has affected the animals that live here. Unfortunately, these examples are only the tip of the iceberg. I could give you 100 more. So the question comes, when considering the animals that such destruction impacts, the question is it wrong? And if it is wrong because it's destructive? Or is it wrong because these environmental modifications, both intentional and unintentional, impact humans? Many of the ethical theories that I've looked at really can only argue for considering the wild animals' welfare in the context of human welfare. For example, utilitarianism is based on maximizing happiness and minimizing suffering. So you can argue for animal welfare by arguing increasing animal suffering is also bad for humans. Or, as some philosophers have done, you can try to include animal suffering in your calculation. But unfortunately, this means that animal welfare falls under the same problem that human welfare has with utilitarianism. When a larger group would benefit greatly from a smaller group suffering, utilitarianism dictates that the smaller group suffering is acceptable. And we tend to underestimate animals, both their numbers and their suffering, because we don't have very many interactions with them, especially in this day and age. When you look at other philosophy, you can find the same problem. For example, Kant focuses very heavily on human interactions. His humanity formula dictates that we ought to treat other humans not merely as means, but as ends into themselves. This is about respecting humans' autonomy. But this excludes animals largely because they're not conscious and independent in the same way that humans are. So in Kant's philosophy, that means that animals have to be shoehorned in under the guise of treating animals poorly also impacts humans. So the question is, how can we account for wild animals in our moral frameworks? I don't really have an answer, but although Leopold gives a glimpse of what that might look like, if you aren't familiar with him, although Leopold was a conservationist who wrote in the early 1900s, he wrote a book called the Sand County Almanac, which frames our responsibilities to animals as a matter of treating them as plain citizens in the larger community. This will allow for some measure of respect without overly favoring animals or humans, but this doesn't really address this fundamental problem in philosophy, that there really isn't much of a framework for considering wild animals within these philosophical frameworks because they're so focused on humans. But if the number of biology classes I've taken over the past few years have made anything clear, it's that history has shown us that we need to consider animals in our frameworks because we've had so many different interactions over the years and impacted them so profoundly that it's irresponsible not to. Does anyone have any questions?
Thank you, Anna. We'll wait just for a few minutes for questions to pop up in the chat. I will share with you that my family owns some property in Estill County, Kentucky, and we have American chestnuts on our property, still growing and thriving. Some of the very few that are left in Kentucky.
That's impressive. Most of them have been hit by the blight, and if you find them, they're all stunted and like half dead.
These are still on a side of a mountain, and I think that maybe because they were covered with other foliage that somehow they survived. So Dr. Cooper's this nice presentation. So do insects suffer insecticides and slow death? How do we know when we go to lower neural developed animals? And it's an interesting topic and difficult to address. Nicely covered topic. That was his comments. Do you want to answer his question?
I spent a lot of time discussing this with Dr. Kirchner, but I don't think we ever came to a conclusion. I'll leave that to people who understand insects' brains a little bit better than I do.
So while we wait just a few more minutes, so tell me how you got involved in research on them.
Well, the honors program has a requirement that you conduct and present some research, and I've done a fair bit in the biology department, but none of it quite met their standards. So I decided that since I was going to need to do another project, I wanted to do something a little bit different because I really like the change in pace that philosophy gives. It's a really different way of thinking than biology has. So it's a nice contrast, I think.
Good. Good. What a great opportunity to have so many choices here at the university. It's great.
Mm-hmm.
So Jessie has a question for you. What led you to this project and topic?
Well, I've taken a lot of biology classes, obviously, because that's my major. But there isn't, like, you discuss philosophy as it applies to conservation a fair bit in those classes, and at least classes that involve any measure of conservation or conservation biology. But there isn't really a standard for how we consider animals, which I always thought was kind of interesting. And a lot of the arguments that you end up having to make are really focused on humans, which just seems a little bit short-sighted. It's like, is it wrong to destroy something because it hurts other humans, or is it wrong because it's destruction?
Right.
So it seemed like it was missing this huge chunk of the picture. And of course, this project didn't really resolve anything, but I think it did put nice bounds on where the problem was.
So, and Dr. Cooper follows up by saying it is difficult to address. I try to be nice to fruit flies as well as crayfish. They are my pets, but also try to treat humanely. All right. Well, thank you so much, Grace. And I looked right at Grace's name when I said that, for being here this afternoon, and to all the panelists. You've done a great job, and we appreciate you participating in the 15th annual Showcase of Undergraduate Scholars. You guys have a great afternoon. I do believe the next session starts, Jessie, at 4:30. And we have presentations from engineering. So if you want to stick around, you are welcome to join us. Thank you.